Okay, so you may or may not have heard of CRISPR. It was this new invention a few years ago. And all this genetic engineering we're talking about, everything we've talked about in genetics, everything we talk about in stem cells, any genetic engineering, you need to be able to like cut out the DNA you want to move and do that. And what CRISPR is a really fast and cheap way to cut and paste DNA. So you can do all this genetic engineering that used to take like so much time. It's really cheap and really fast. So it's a really cheap, fast way to cut and paste DNA. And it has just opened up the world to so much more gene editing and stem cell use and genetic engineering. So this video is like so awesome. I think I don't know if it's by Ted Ed or whatever, but it really, really explains it really well. So if you want to watch that, you can. All right, so CRISPR is really good at cutting and pasting your DNA. How does it work? Well, we figured out, hey, bacteria are really good at becoming immune to viruses because what they do is they cut out the DNA of the virus and they keep it as like a keepsake to remember that virus so that it can't hurt it next time. So we're like, oh, bacteria are really good at cutting DNA. So basically, we use that. We use bacteria. CRISPR is made from a bacteria. And we're like, hey, you're cutting DNA, we're going to program you to cut the DNA that we want you to cut for us. That's a simple definition. If you want to learn more about it, you can watch videos. But what can we do with it? So we can cut and paste DNA to cure disease. Like we could use CRISPR to cut out unhealthy DNA and replace it with altered healthy DNA. We could cure genetic disease. Huntington's, muscular dystrophy, or sickle cell anemia. The downside is that it would be expensive and difficult in these diseases. Remember, your DNA is in all 37 trillion cells of your body. You would have to change the DNA in all 37 trillion cells in your body. So that's a huge challenge. So what's a better alternative? Well, let's edit the DNA before you're made of 37 trillion cells. Let's do it in like little eggs and embryos and like little developing babies before there's like a trillion cells to edit. So that's what we have also been doing. Genome editing in embryos that are implanted in the mother. So again, I said women who are having trouble getting pregnant or whatever, there's some problem, they take the egg out of the mom and the sperm out of the dad and they put them together like in a test tube. That's why they would call them test tube babies. Get everything fertilized and then implant it into the mom's uterus because for some reason they're having trouble. While you're doing that, you could go into the egg and like cut out or go into the sperm. I'm not sure if they've done that. And cut out any bad DNA. So you could prevent these diseases before they happen in 37 trillion cells of your body. So that's the plus side. The downside is, are people going to be like, hey, while you're at it, could you make my baby really smart and really fast and have like perfect vision and have like blue eyes? So we don't want what we call like scary designer babies. And that's where in class we would watch the movie Gattaca, and it is so amazing. The premise is that only the rich can then afford designer babies. So then all the rich have like all these great genetic traits, and the people who can't afford it maybe don't, or maybe they do just naturally, but they're discriminated against. So like when you go for a job interview, they don't even look at your resume, they just look at your DNA. So it's kind of, and they call it, instead of racism, they call it genism. So it could be like unequal access, like rich people whatever they want and kind of rule the world. So that's a scary thing. But we could also use it to cure disease, right? So another thing we could do with it is you can take out, it's called immunotherapy. You take out someone's immune cells and you use this gene cut and paste editing of CRISPR, and you use it to program that cell, that your immune cells to kill the cancer cells. So right now we use like chemotherapy to just kind of like kill a lot of cells and it makes a person sick. It's supposed to kill the cancer cells, but it kills healthy cells too, and it makes the person immune system cells to target like just those cancer cells. That would be like really good. And you could also make it personalized. So it's like you're using just the person's actual cells. And the downside is always that it's so difficult to figure this out, and all this stuff is really expensive. We can use it to study diseases. So sometimes the best way to see what a gene does is to like cut it out what goes wrong. So they used to do that with brain injuries. Like if someone was missing this part of their brain in the back of their head, they couldn't see. So that's how we were like, oh, that's the occipital lobe. That's the lobe of your brain that helps you see. And if they lost like this part, they had trouble with hearing. If they lost this part, they had trouble with decision making. So sometimes like taking something away is how you figure out what it does. So that's how we figure out what different genes do. We take them away and then we go, oh, this person they don't code for eye color anymore. That must be eye. It's a good way to study genes or diseases. Okay, so gene drive is kind of a crazy thing. I think this video explains it the best if you want to watch. I think it's this 2-minute one, but basically like to get rid of all the mosquitoes in the world that are carrying malaria. So you create these little mosquitoes that can't carry malaria, but then you let them out into the population and they don't like take over. Like there's now you have some mosquitoes that can't carry malaria, some that do. So what you can do is you can like program those non-malaria mosquitoes to like take over, so that when they get together and they mate with a disease carrying mosquito, they can like force their genes in baby mosquitoes that can't carry malaria are born. It explains it really well in this video if you're interested. We've also used CRISPR, and before CRISPR, just slower, older gene editing to alter plants and food. So we've made like food that's drought resistant, or food that has more nutrients, or food that's resistant to pesticides, or food that can't be certain bugs or insects. So in that way, we've created a lot more food, a lot more productivity. It's good for farmers. The bad side is that maybe those genetically modified foods, organisms, GMOs, are bad for us. And you can look more into that if you want. Okay, and then the last thing about CRISPR is that all the stuff we've been talking about, stem cells, and programming them to become different things that we need, helping them make us a new heart or a new liver. All that stuff can be done better now with CRISPR, because CRISPR is just this way better, way faster, way cheaper way to cut and paste and edit DNA. So the upside is that, I don't know, you could cure like every disease ever by going into the DNA and cutting it out. The downside is that you could have some unintended consequences and maybe you cause something bad. And we'll talk about that in our next video, which is risks of stem cell therapy. Just showing how much CRISPR has been used. So I think it was developed in like 2007 and it was being used a little bit. And then as soon as scientists figured out, this is how many times it's been cited in like a research paper by scientists. And it's just totally, just like everybody's using CRISPR now. Any regular old scientists, not even like the super advanced ones, are using CRISPR to do any of their gene editing that they need. So it's really revolutionized the gene editing industry. Okay, so just to conclude, CRISPR is a faster, cheaper way to edit DNA, cut and paste it. So we can use that for any of the gene editing things that we've talked about in stem cells or in cloning or of training bacteria to make insulin for us. All the gene editing, all the genetic research we're doing, we can do it faster and cheaper with CRISPR to cut and paste it.